Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator at Simulations Plus. I'll be hosting today's webinar behind the scenes, making sure we don't have any technical difficulties. In this webinar, Dr. Viera Lukacova, Chief Science Officer of PBPK CHEM Solutions, and Susie Zhang, Vice President of Regulatory Strategies, will outline a reliable mechanistic PBPK modeling framework for predicting CYP enzyme-mediated DDIs with a high degree of confidence. Our Business Development Director, Andy Mueller, will serve as our moderator today. A few housekeeping items before we begin. We take your privacy rights seriously by registering for and attending this event or participating in the Q&A session. You are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address all the questions in the Q&A after the presentation. And before we begin, I'd like to conduct a short poll to understand your familiarity with DDI risk assessment. And I'll go ahead and launch that now. And we'll give everybody a couple seconds to answer here. Answers coming in. Okay, and I'll go ahead and end the poll there. And I'll pass it over to you, Andy.
Thank you, Jasmine. Hello, and welcome to today's webinar, Mastering DDI Risk Assessment, Prediction of CYP Enzyme-Mediated DDIs with Confidence. This is our second in a series of PBPK-focused DDI webinars that we'll be offering in the coming months. Thank you very much for taking time out of your busy schedules and joining us this morning. I'm Andy Miller, and I'll serve as your moderator. I've spent essentially my entire career in pharmaceutical development, and my scientific background is in toxicology, bioanalytical chemistry, pharmacokinetics, and PBPK modeling and simulation. I currently serve as the Director of Business Development for PBPK Services, QST Services, and Regulatory Strategies here at Simulations Plus. I'm very pleased to be joined today by my colleagues Susie Zhang and Viera Lukacova. Susie brings to us nearly 13 years of experience from the FDA, in which she focused primarily on PBPK modeling and clinical pharmacology. During her time at the FDA, Susie oversaw and reviewed many PBPK DDI submissions, which truly makes her an expert in what the agency expects in terms of DDI investigation within the development pipeline. Viera is the Chief Scientific Officer at Simulations Plus. She's been with the company for about 15 years, working on the GastroPlus, DDD Plus, and Membrane Plus software packages, as well as contributing to modeling studies, helping companies with their drug development programs through multiple stages of drug development, processes, and interactions with regulatory agencies. I've prepared a series of questions for Susie and Viera. While they take time to respond to my inquiries, I welcome you to post your own questions in the chat. I will be sure to reserve some time at the end of my questioning to allow for questions from you, the audience. With that, welcome Susie and Viera. Would either of you like to say a few words before we begin? Susie, maybe?
Sure. Thank you, Andy, for the introduction. Hello, everyone. As Andy introduced, I have background in clinical pharmacology and pharmaceutical science. I worked at FDA for over 12 years. Currently, I serve as the Vice President of Regulatory Strategy at Simulations Plus. Here, I'm devoted to help clients to explore their products to go to the market affordably. And I look forward to today's webinar. Viera, over to you.
Thanks, Susie. Hi, everyone. I'm looking through the list of everybody who joined, and I see a lot of familiar names. So thank you for taking time, and please do make sure to pose any questions that you have. This is what the webinar is about, to make sure that you do get answers to your questions. So looking forward to this discussion with Susie and Andy, and also all of you online.
Excellent. Thank you, Viera. So we can get to the questions and presentation. Before we begin, I would like to mention that we are recording these presentations. If you were not able to attend the first webinar, please do feel free to reach out to me directly, and I can connect you with that recording. This is the second in the series. So if you missed episode 1, let me know and I can get you going with the series for some binge watching. Question 1 for Susie and Viera. So during our first webinar, we discussed both the M12 DDI guidance and the M15 MIDD guidance. And I have a follow-up question on that topic. Since the publication of the global M12 DDI guidance, which overrode the previous regional guidances, what are the key takeaways from the M12 DDI guidance within the context of enzyme-mediated DDIs in particular? Big question. Sorry.
Thank you, Andy. Yeah, thank you, Andy. Maybe I can take on that question from a guidance perspective. So M12 guidance, let me see. So as you can see, M12 guidance is more of a technical guidance, and it included enzyme-mediated and transporter-mediated drug-drug interactions. That's the main mechanisms included in that guidance. And in terms of modalities, it includes small molecules, monoclonal antibodies, and antibody-drug conjugates. For the latter two, there are also FDA published guidance dedicated for those modalities, and also covered methodology in terms of in vitro evaluation, clinical evaluation, as well as in silico evaluation. Now coming onto the enzyme-mediated drug-drug interactions, I really think that we should narrow down to each enzyme. We need to study enzyme by enzyme. When we talk about drug-drug interactions, we talk about two parts. One is the drug as a substrate, and the other is the drug as perpetrator. And for the drug as a perpetrator, we need to evaluate its inhibition potential as well as induction potential. Now let's look at them category by category. So first is to evaluate the investigational drug as a substrate. In general, it is more complicated to evaluate the drug as a substrate, as you need to understand how the drug is being eliminated. Let's look at the enzymes that are covered by the guidance first. I put them into a few categories. So the first one is the common CYP enzymes that you see a lot. For this category, the major enzymes included are CYP1A2, 2B6, 2C8, 2C9, 2C19, 2D6, and 3A. Each enzyme has its own unique properties. For example, if your drug is a CYP1A2 substrate, you probably need to think about the smoking, the impact of smoking. As we know that smoking is a weak to moderate induction inducer of CYP1A2, and in your clinical trials, do you have smokers enrolled? If your drug is a CYP2B6 substrate, and the issue is there's no known strong index inhibitor, so how are you going to evaluate DDI if your drug is a CYP2B6 substrate? And for CYP2C9, for example, we know the enzyme has genetic polymorphism, so you probably need to think about how to evaluate the impact of genetic polymorphism in addition to DDIs, and how those two factors combine together to impact the PK of your investigational drug, and similarly to C19 substrate and 2D6 substrate. And for CYP2C8, 3A substrate, this is the most commonly seen category where your investigational drug is a substrate of CYP3A. And for this enzyme, often we see that the drug may also be an inhibitor and inducer of CYP2C3A. Then you will have autoinhibition and autoinduction potential. And another common issue with CYP3A substrate is it potentially be a dual substrate of PGP and CYP3A. So when you design your clinical trials, even modeling simulation, how are you going to separate these two pathways? And I'm sure you will be asked to evaluate the impact of each pathway separately and in combination. The second category is a phase 2 enzyme. You'll see a lot of UGT enzymes here. So when the investigational drug is a substrate of UGT, you probably need to consider the potential pharmacogenetic effect. And the other challenge is the inhibitors that we are going to use to study the effect on those drugs or enzymes may not be specific. But the good news is the interaction magnitude usually is limited. And there are other CYP enzymes, other phase 1 enzymes, and other phase 2 enzymes. For those enzymes, the challenge is there's no known specific clinical inhibitors or inducers. So overall, to understand the ADME of your investigational drug and it's important for the further drug-drug interaction evaluation. And also to use PBPK to evaluate the DDI, validated probe inhibitor and inducer models are the key. And we will talk about that a lot later on. When you evaluate your investigational drug as an inhibitor, so again, we look at enzyme by enzyme. So for some of the enzymes, the challenge is that it may be a mixture of inhibition and induction effect. So how are you going to evaluate that? How are you going to design your in vitro study, your modeling and clinical trials? Oftentimes we see that a CYP3A substrate, again, also a inhibitor and inducer of 3A. So that is considered as a complicated drug-drug interaction case. But overall, this is an area where PBPK modeling has high confidence. Again, to have high confidence to evaluate your investigational drug as an enzyme inhibitor, validated sensitive probe substrate models are the key for successful evaluation. Coming to your investigational drug as an inducer, so in general, according to the guidance, you evaluate first 3 enzymes, 1A2, 2B6, and 3A4 to cover different mechanisms. And when your drug is demonstrated as a CYP3A4 clinical inducer, you need to further evaluate the induction potential or induction effect on CYP2C enzymes, as they may oftentimes they are co-induced, and also induction potential on UGT enzymes, as well as the effect on oral contraceptives. What we've seen is the second part is often missing by the sponsor. So yeah, that's the key takeaways, I think, from the M12 guidance. I'll give it back to you, Andy.
Excellent. Thank you very much, Susie. Appreciate it. So the next question I have is, how can our partners assure confidence in DDI assessments and ultimately gain regulatory buy-in when leveraging PBPK modeling approaches for DDI simulations and evaluation?
Yeah, so again, I prepared a couple of slides, as you can see here, and we also discussed a little bit in previous slides that, so there are some general considerations. For example, there are always model assumptions, and are those assumptions adequate? So we need to provide justification for model assumptions, as well as PK parameters or physiology parameters that being used, even physical chemical properties of the molecule. The resource of those parameters, if it's optimized, are they reasonable? And also need to include in the report the content of use of the model structure, the development plan, model validation, how those are validated, those type of things. And importantly, the sensitivity analysis should be included in the PBPK analysis. So these are all in the guidance. There are some specific considerations for the investigational drug as substrate. For example, whether the model could describe the available clinical PK data using different dosing regimens. This includes not only the PK data for the clinical or recommended phase 2 dose, but also some other available doses, especially lower dose levels, because those will tell you whether the model has captured the potential mechanisms involved in absorption, distribution, elimination correctly. if you don't want to include those, then justification should be provided. And what's important is also the validated perpetrator models that you are going to use to evaluate your investigational drug as substrate. And most importantly, the confidence in allocation of your clearance pathways. For example, you want to allocate 50% to CYP3A4 and 20% to CYP2C9. Are there enough data to support this allocation? To evaluate your drug as a modulator, what's important is, again, the model could describe the available clinical PK data, all the available data, and you need validated sensitive substrate model, as well as some sensitivity analysis and justification for the range. So that's the consideration. And what's also important is the considerations for metabolite as early as you can. These are the criteria listed in the guidance, but what I wanted to point out is to provide justification if you want or don't want to include metabolite in the model as early as possibly can in the development program. So for example, when considering whether a metabolite should be included as a substrate, the guidance says a change in metabolite exposure can result in clinically meaningful alteration of efficacy or safety of a drug. If that's true, then you should include the metabolite in the DDI considerations. However, there need data analysis to support either way, whether it's clinically meaningful or not clinically meaningful. There you probably need some work. Similarly, to include metabolites as an inhibitor and inducer, there are detailed criteria in the guidance. So I would say there are some work need there to justify either way. But my suggestion would be to make a decision as early as possible. And I'll let Viera to talk about some case examples. Viera.
So this is one of the case examples that was actually just recently published. And many of you who have worked with our team on developing the models for your own products, for your own pipelines, you would know that this is basically the process that we follow and that we recommend, right? The first thing is really think about the model structure. Include all the mechanisms that are critical for the to account for the compound's own PK, as well as for the possible impact on other compounds, right? And this is the model for carbamazepine, where we modeled both carbamazepine as well as its metabolite, right? And here I just wanted to make a quick note on the metabolites, right? So there is, as Susie was highlighting in the previous slide, there are certain requirements from the regulatory aspect, right? In which case the metabolites need to be considered and need to be accounted for. Even aside from what regulators might require, right? Considering the metabolites can be useful also to have a better validation of the model, especially if you are dealing with multiple pathways, right? If you do have those metabolites measured, and if they are contributed by different enzymes, accounting for them in the model can help you to better parameterize or to better distinguish how much each pathway is contributing to the PK of the compound. So again, come back to what the point that Susie was making, decide early on including or not including the metabolites, but think about it from the point of not just the check boxes on the guidance, right? But also from the point of what are you trying to achieve with the model, and where that metabolite could provide additional information, could provide additional help with your model. And that's exactly what we've done here. What you see in that middle section with the four plots, goodness of fit plots showing the AUC predicted and observed, and Cmax predicted and observed for both carbamazepine and and the metabolite of the carbamazepine. And it may be the legends within the figures may be a little too small, but you would see that here we were looking at different administration routes, IV, oral, different types of formulations, solutions, suspensions, tablets, right? And both single and multiple doses, right? So again, making that point, use all the data that you have available to help you validate the model, right? The more you can describe from your clinical data with the single model, the more confidence you have that the model is capturing all the critical mechanisms. And the more confidence you have, the more confidence the regulator will have that the model actually is correct, that it is capturing what it needs to capture, and they can rely on the model to make the decisions, right? And finally, if there is already DDI study, if you might have already performed DDI study with some compounds, then that would be the final set of validations, right? To help us distinguish or to look at whether the model is able to reproduce those existing DDIs. And that gives us more confidence as to how it's capturing either the impact on the enzymes or the contributions of different enzymes to that pathway, right? And I think this is the question that we often get asked, what are the criteria for the accuracy of the model that we are looking at, right? And when it comes to the PK, we do try to, so the Cmax and AUCs, we do try to get within the bioequivalence limits, right? So within 25%. But it's not strict criterion. At times you may have a compound which has a lot of inherent variability, right? So bigger errors we might accept, but again, it's something that you need to be able to justify, right? Why the compound may have bigger errors of predictions on certain parameters. And when it comes to the DDI predictions, I think this is now fairly widely accepted and commonly utilized using this criteria proposed by Guest and colleagues, where the acceptance criteria depends on the magnitude of the effect. So if the effect is small, it's close to 1, then the acceptance criteria would be very small, only within about 25%. If the effect is big, then we would be accepting higher prediction error on the DDI.
Yeah, thanks, Viera. And Andy, I hope we addressed that question in terms of developing confidence in PBPK model prediction.
Yeah, excellent. Yes, thank you very much. Thank you both. So follow-up question to that. Although today's webinar is primarily focused on the CYP-mediated DDIs, which are potentially the most common, the most well-known, are there other categories of DDIs to take into account? And if so, what other types or categories of DDIs can be simulated within GastroPlus and BiSimulations Plus?
Yeah, Viera, do you want to take on that question?
Sure, I'll take on that question. Yeah, so the simple question is any that you need. So this is highlighting, and I'm not going to go through a lot of examples. There is a lot of them in literature. Here is just showing few that were actually used in the regulatory applications. But of course, the enzyme specific DDIs. And today we are talking about CYPs, but it has been also used for UGTs. So there are aspects as well, if you go back to the previous slide as well. So the enzymes, right, the transporters as well. This is where there is still a bit of uncertainty, but the transporters are utilized. And we will have another seminar specific on the transporters, where you will learn more about the models that are available and how we validated them. So I don't want to spend too much time on that. But these are just a couple of examples here showing from publications where the companies highlighted the use of GastroPlus on the predicting the transporter-mediated DDIs as well, and how it was viewed by the regulatory agencies. And finally, on the next slide, the part which is becoming more and more utilized, even though it's still sort of not the first thing that people think about when they think about DDIs, right? But it is a DDI. It is a more of a PD-mediated DDI. This is where we are talking about impact of acid-reducing agents on the PK of your compound, right? A lot of people, a large portion of the population is taking some sort of acid-reducing agent, so you need to know how it's going to impact the PK of your compound, or the changes in the gastric emptying time, right? If it slows down the gastric emptying, again, how it's going to impact the PK of your compound. And all of these can be also accounted for in GastroPlus. And this was one, a few years ago now, a nice publication from the several different contributors from different companies showing the applications on the acid-reducing agents or pH-dependent drug-drug interactions.
Including our very own Neil Miller.
Yep.
All right, thank you. So great message. So far beyond just CYP-mediated DDIs can be simulated in GastroPlus successfully. So with that, with knowing those full set of capabilities, how can GastroPlus easily help our partners in the industry facilitate simulation of DDIs to inform their label? So the tools are clearly there. How can we make it easy and accessible to our customers?
So in general, right, as we've already discussed, you can do any DDIs between any compounds, right? All you need are the models. The part where we are making it easier for you is that we are providing the models that we have already built, right? And the models that we are building are really the ones that are the most common, the ones that you typically do need to use, and covering a wide variety. The inducers, if relevant, inhibitors, different categories of inhibitors, weak, moderate, strong, right? And substrates, right? And what we are highlighting here is we are really trying to do the cross-validation, right? So when we validate a substrate model, it's not just with one inhibitor and we are done, right? If we build a substrate model, we try to validate it with as many different inhibitors and inducers as we can, right, based on the data available. The same comes to the perpetrators, whether it's inducer or inhibitor, always validation with multiple different substrates. So, and as you can see in these networks, these are highlighting some of the networks that we have available in terms of how the models were built and cross-validated for different mechanisms, right? So again, this is something that we provide to our users of GastroPlus. Clearly, they are models that are working in GastroPlus, so that's who would have the most benefit out of those. But and again, here I want to stress that these are not the only models that you can use. You can use any models, but these are the ones that we already created, so we are providing them to our users as a convenience, so they don't have to keep redoing the same work that we have done already. And since we are talking about the CYP3As and validations on the next slide, these are just few of the examples. Of course, you've seen on the previous slides, well, for the 3A4s, we have wider range of models. So this is showing just some of the validations that we are using, right? And for the first two, for itraconazole and voriconazole, the first two plots, the top two plots, are showing the validations of the PK. Right? As I was mentioning earlier, the first critical aspect is every model needs to describe the compound's PK. And this is where we really look at the PK parameters of the compound itself across a wide range of doses, formulations. And for itraconazole, you see that those two plots are a little bit more colorful because we were looking also at metabolites, right? So it's not just about itraconazole, but also its metabolites. So everything that is critical is included, and we really try to make sure first that the PK is captured well. Right? And then we look at how well the model is predicting the DDIs. Right? And again, it's validated, each of these models is validated across multiple different substrates. And depending on how many studies we can find in the literature, you would see some of these plots, have more or fewer data points. I think rifampicin in the upper right corner as an inducer, that's where we were able to find most of the DDIs in the literature. So very rich plot showing very nicely predicting DDIs with different compounds. Fluconazole, right below that, again, it's showing a more colorful plot. This means different substrates, looking not only at 3A4s, but there are also 2C19 and 2C9.
2C9.
Yeah, 2C9 and 2C19, right? So again, the important aspect here again is we use all the data that we can find, right, and try to cross-validate with as many studies as possible, and always with multiple compounds. So really providing the models that you can then use with confidence to predict the DDIs with your own compounds.
Yeah, thanks, Viera. I think that stressed the importance of cross-validation when we develop these probe substrate models, as well as modulators, inhibitors, and inducers, that we use the same set of parameters and then cross-validate against all the available substrate that we have, those models. Is that all we wanted to address for this question?
Yeah, I think somebody posted question just now, so maybe I can address that one in Q&A right away. There was a question that some of the points are outside of even the twofold limits, right?
Mm-hmm.
This is something that we are addressing. Let's say if you look in the itraconazole model, there are few points that are in light points in the bottom plot, right, for the DDI predictions. Those are the ones that we showed, and this is something that you will get complete report explaining all of these figures, where we explained why those are not reliable, and when we adjusted the model, right, that it falls more within the lines, right? There are also some predictions where it's simply variability, right? For example, if you look at the rifampicin, right, there are in the upper right corner, there are a couple of points that are higher than the red lines, right? This comes from the study which was based on midazolam, based on the study design. It was the same as all of the points that fall within the lines, right? And yet they've seen different prediction, different DDI in that specific study. So some of these simply come to variability between the studies, which, if everything else, if there is a number of other studies with exactly the same compounds, with exactly the same design, and they fall within one range and one study is outside, it's just an outlier that in some cases we may not be able to explain. Right? So it's all about as much as we can explain with the model.
So yeah, so again, these are the plots from the report, and you will be able to get the reports. Are there any other burning questions in the Q&A? I cannot see it.
I don't see anything.
No, just the one from Vicky, and thank you very much, Vicky, for posting that. Please keep those questions coming. We're happy to attempt to address them in real time. If we can't address them in real time, we do have time for Q&A at the end. But thank you very much for the question, and please do ask more. So a follow-on to what was just presented. So our partners in the industry that may be interested in doing PBPK modeling for DDIs, can you speak to how our partners can access these models and how we can support them and attach them to these tools?
Oh, yeah, sure. I will briefly show the screenshot, and then Viera will show you how to get those models as well as report. Currently, we're continuously uploading our models into our learner pond. So this is our learner pond where we store all the training materials, webinars, etc. We are also uploading our DDI standard models as well as reports into the that website. And after you log in, you go to resource, and you will be able to find all the files as well as model files as well as reports there. If you see the tag as archive, there are the zip files, and those Those zip files include all the modeling files. And if you select a document from the type, you will see the documents here, and those are the reports. I'll transfer the sharing function to Viera so that she can show you how to actually do it.
So this is just a quick live screen of what Susie was just showing on the slides, right? When you log into LearnUpon, and again, this is something where you can register with your company email, and you will have access to this resource. And the beauty of this is you don't have to keep asking us for the models anymore, right? I know that many of you have worked with our models in the past, and you got used to sending us email, hey, can you provide this model to me, and so on. You won't have to do that anymore. You have those models and the reports available on the LearnUpon, right? So when you log into the platform, as Susie mentioned, under resources, you will have this large list, and it's several pages long because the models are growing, and we already have, I think right now there is about 20 models with the reports, and there is more that will be coming in next few weeks. And again, the archives are the models, the actual GastroPlus models that you would load in the GastroPlus and do the predictions. And when you change the type into, let's say, document, right, these are the PDFs of the reports describing the model development. Each of them describes the model development, how the model was validated, all of those validation results, and so on. So these are really ready reports that you could submit to the regulatory agencies if you are using it for the regulatory applications, or just to get the feedback or see the details of the model, how it was validated, which mechanisms it accounts for, and so on. So this is hopefully something that will streamline your use of these models, since you have access to them at any time. And it also makes it easier for us to keep uploading new models as we keep building them. So this is something that will be updated on ongoing basis. Every time we have a new model, it will be automatically added. And that being said, it doesn't mean that we don't want you to contact us anymore, right? If you come to the site and there is a model, there is a report that you don't see it, that you would like to have, make sure to reach out to us, right? It might be the model that we are just finalizing and maybe coming in next week, right? Or it may be a model that we haven't done yet, simply because we are prioritizing based on what you are asking for. Those are the most critical for your development programs. So if there is a model that you don't see yet, let us know so we can start looking into potentially building that model and start prioritizing it. Okay.
Thanks, Viera. Are there any questions on this?
Let me
Not
check the
specific.
Q&A.
Yeah, general questions which I think we can answer in Q&A at the end.
Mm-hmm.
Not specific to this.
Okay. Let me share my screen again. Okay.
All right, are you ready for another question?
I think so.
Okay.
If you have any more questions, sure.
I've got a few more. High level question, sort of breaking away from the specifics a little bit. On behalf of our partners who are considering incorporating DDI PBPK modeling into their development programs, when is it appropriate to start thinking along those lines and maybe implementing those modeling activities?
So I'll take that question again, Susie, if you can go, and you may need to do a couple more clicks. So I'm going to take that question more broadly as to not just DDI PBPK models, but the DDI evaluation in general, right? And this is something that you should start thinking about very early on, right? Even in the discovery stage, using machine learning models, and some of those are highlighted here from ADMET Predictor that we are providing, right? where you can already start getting predictions on if the compound is a substrate or inhibitor of a specific enzyme or a transporter, right? For some of those, we even have models for Km and Vmax values that you can directly use in the model for PBPK later on, right? But even very early on, even just the information as to which enzymes, which transporters may be impacting your compound, or which may be impacted by your compound, can be already something that you might be using to decide on the compounds, right? To start looking at what is that potential DDI liability. And if you have to make the decision between, from number of compounds, maybe you could lean more towards the ones that it is going to be less, right? So this is the decision that can help you in that early stage in the evaluation, just what might be happening in the future, right? And of course, as you are going in the initial evaluation, start doing the studies for the compounds that you selected, start doing the in vitro measurements. You may have data, you may not have enough to build a full PBPK model, right? But you may already have some initial information on the in vitro FM values or quick in vitro screening data for the inhibition induction parameters, right? And this is where you can start utilizing those static equations to start predicting the potential DDI, right? And this was, again, one paper that is a few years ago. This group created a nice paper of highlighting how this strategy can work, right? And they've used it specifically for time-dependent CYP inhibitors, how they would use the in vitro data to screen, or the in vitro screening data to start predicting early on that potential. But the same strategy could be applied for other types of applications, right? So the DDI in general, you can start and should start looking at as early as you start thinking about your compound. And of course, on the next slide, as you get to, as you proceed with your project and you start generating more data that allows you to build a full PBPK model, this is where, and this is again one of the examples, this was from a actual development project, where all of the same process I was describing earlier, right? Once the PK data was available, it was used to build model, validate to make sure that it's capturing well the PK as well as the existing DDIs, right? Then it was linked with, for the validation of the existing DDIs, the standard models were used, right? And other standard models were then used to predict the DDIs that were not tested yet. And that's that table in the bottom right corner, right? Because that's why you are building these models, right? You are not building these models just to reproduce what you already measured. You are building the model so that you can start predicting what you didn't measure yet. Right? So that would be the progress there.
Thanks, Viera. Completely agree that DDI assessment should start as early as possible in the drug development program. Although you may start clinical studies later, but those assessment, at least in vitro, in silico, should start as early as you can.
And maybe even using tools outside of GastroPlus, such as AdMet Predictor, some of our AI machine learning tools to start evaluating during early development, lead optimization, discovery processes. Excellent.
Okay, all right, thanks, Viera.
The next question I had may actually have maybe playing into the question that's in the Q&A right now, and sort of back to more specifics. Are there any key considerations that should be taken into account when multiple pathways are involved in DDI interactions?
Yeah, I can take on that question. So when multiple pathways are involved, the things get complicated. One pathway probably only need to consider one pair. Two pathways, you won't be doubled, but maybe tripled in terms of considerations. But again, when multiple pathways are involved for the investigational drug as a substrate, it's more complicated. Or initial assessment, you can do is to do it by some modeling simulation approach to determine the appropriate studies. For example, you need to do some in vitro, in vivo extrapolation to determine which is the mean pathway, and the extent, to have some ideas before you even start the clinical study. And the studies need to be carefully designed if you're going to do clinical study, and maybe one or two or more. But the purpose of the study need to be specified, whether you want to evaluate a specific pathway or evaluate an overall effect as a worst case scenario, whether there are any potential confounding effect in your clinical trials, and how many pathways you're going to evaluate in one study or in multiple studies. Those are the questions that need to be considered even before the start of clinical studies. And also, how are you going to use PP model to tease out each effect or to evaluate the combination effect? If you need to evaluate the combination effect, what data you need to get before you even do the study, and what is the optimal clinical trial design? Those are some of the considerations. And then when applying the PBPK modeling approach to evaluate which pathways, which perpetrators, how many concomitant medications will be evaluated simultaneously. So those are also some of the questions to consider. What are the commonly used concomitant medication for a specific indication? Those type of questions. To evaluate when the investigational drug, when multiple pathways are involved for the investigational drug as a modulator, similarly, each pathway should be evaluated with a sensitive index substrate so that it's clear on each pathway. And then you will also need to consider when the commonly used concomitant medications are potentially impacted by multiple pathways from this investigational drug. How are you going to evaluate that if the DDI studies or in vitro studies only evaluate each pathway? Will you do some modeling simulation to evaluate, which I think so. That's where the value of modeling simulation is, to evaluate some of the scenarios that you just cannot test each of them in clinical trials. So I think those are just some of the general considerations. For each program, you probably need to consider a little bit more based on the properties of your investigational product. That's my general thoughts for the question about multiple pathways.
Excellent. Thank you, Susie. That concludes the questions that I had prepared for today. So we are going to open this up to Q&A. Before we do, what do we have chambered for upcoming webinars in the series, Susie? So we've the first webinar was really focused on high-level regulatory aspects and requirements for DDI modeling and simulation. Today we covered primarily the CYP-mediated enzymes. What's on deck?
Yeah, we'll be talking about transporter-mediated DDIs, which is also large portion of DDIs in April. And then in May, we'll talk about PD-mediated or absorption-related DDIs, for example, elevated gastric emptying time, delayed gastric emptying time. Actually, we separate food effect, but you can consider food as another extrinsic factor that may impact your PK, investigational drugs PK, via absorption process. And in September, we'll be talking about complex DDIs where multiple pathways are involved, or transporter enzyme interplay may be involved in DDIs. You can follow up on our upcoming DDI webinars through the website where we have a resource that you can check periodically. So that's the planned webinars for the next few months.
And a little teaser, spoiler alert, we may have a few bonus ones sprinkled in, right? We're speaking with some of our partners in the industry about putting some bonus presentations together and maybe doing some specific case studies, but those are to be determined, but we're working towards that as well. So thank you very much. Those are all the questions I have. I believe Jasmine has a poll question that she wants to launch, and then we can open it up to a little bit of Q&A with the time remaining.
Thanks, Andy. So our poll question is basically asking if you're interested in setting up a 15-minute call with Viera and Susie about DDI risk assessment. And it's a simple yes or no here. And I'll give everyone a couple seconds. All right, I'll go ahead and end it there. And then for those of you that did say yes, we'll follow up. And I'll pass it back to Andy.
Thank you very much, Jasmine. Just one question in the Q&A today from Janik. The question is, hi, would it be possible to optimize a parameter when you have data from multiple trials? For example, can you optimize intrinsic clearance for 3A4 with 2 or 3 inhibitors, possibly in the next version of GPX, possibly in the current version of GPX?
So this
Susie,
is a more technical question. I'm guessing you are asking about how to actually process that or do the optimization. So the first aspect is when it comes especially to the DDIs, right? The way the program can distinguish and look into which pathway or which process is impacted is really through the specification of a given enzyme or a transporter, right? So the first place is you do need to make sure that you set up your KM and Vmax values for that specific enzyme, because that's what tells the program what would be happening. The other question is, not sure if you are asking about the technical optimization, because in general, of course, it's possible, right? If you have the data, you can modify the parameter values. It is not linked to the optimization module yet, the DDI, so you cannot just tell the program, optimize it against these data sets. But if you have tried the GPX, you would notice that even if you have to do the manual modifications of the parameters right now, it really is much more flexible and much easier to do in the GPX than in the legacy versions, where you don't have to keep exiting the DDI module. So just a few changes on your Vmax value, very easy to do these days. So heading in that direction, right? But again, the key aspect to remember is you need to, if you want to do the DDI, you need to make sure to let the program or let the model know which mechanism, which protein, which enzyme or transporter is really impacted.
All right, thank you for that, Viera. Okay, I think that concludes the, did we have anything else pop up into the Q&A just now maybe? Nope, I think we're good. So thank you very much, everybody. I really appreciate you carving out some time for us today. Please do follow us for the additional webinars. Those will be going through early fall. Jasmine was kind enough to put a link in the chat of the previous recording. That recording, this recording, and all future recordings will be available. Feel free to reach out to us, me in particularly directly, if you would like those recordings. With that, thanks again, and I will send it over to Jasmine to wrap up today's webinar. Thank you.
Yeah, thank you all again. For any questions we were unable to answer live today, someone from our BD team will reach out to you with answers following the webinar. We invite you to visit our website to learn more about our DDI risk assessment. This webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you, everyone.
Thank you, Jasmine. Thanks, Viera. Thanks, Susie.
Thank you, everyone.
Appreciate it.
Thank you.